Dear students, our Mdcat biology next lecture is nucleus. Nucleus is very important, and firstly we were discussing the introduction of nucleus. Nucleus is actually the presence of nucleus was firstly reported by a scientist, which is Robert Brown, and it was reported in 1831. Actually, the cell was reported by a scientist, which is known as Robert Hooke, and Robert Hooke discovered the cell in 1665. This is a very important point. And the nucleus was discovered in 1831 by the Robert Brown. So the nucleus stands out in many cells due to its slightly darker appearance compared to the surrounding cytoplasm. This point is actually that when we stain the cell, the cytoplasm look lighter. Its appearance is lighter, but the nucleus appearance is darker. So that is the point when we do the staining. Then the nucleus appears slightly darker, and the lighter will be the cytoplasm. Next is the function of nucleus. Actually, as we know that a cell contains the nucleus into its center, and it controls all the activities of the cell. So it controls the life and activities of the cell. All activities taking place into the cell are controlled by the nucleus. So in case of animal and plant cell, there is a difference into the location of this nucleus. For example, this is an animal cell. So in animal cell, the nucleus is located into the central position of the animal cell. But in case of plant cell, this nucleus is pushed toward the periphery. This is a periphery. So that is due to the presence of this large vacuole. This large vacuole present into the center. That is why the nucleus position is peripheral. So central position in animal cell and the peripheral position into the plant cell due to the large vacuole present into the center. Shape is irregular, sometimes it is irregular, and sometimes it look like spherical shape. Next type of nucleus, as we know the word mono. Mono means single. So nucleate means nucleus. The cell with a single nucleus, it will be called mononucleate. But the cell with the two nucleus, for example, it will be bi mean two and nucleate mean nucleus. So cell with the two nuclei is known as binucleate, as the term is multi mean many, and nucleate mean nucleus. A cell with the more than two nuclei, these are multinucleate. For example, muscle cell. So human muscle cell contain the more than two cells. These are the multinucleate cells. Visibility of the structure of the nucleus. Look, the nucleus is only visible when there is non-dividing phase. When there is a dividing phase, during the dividing phase, as we know, for example, this is cell membrane, and this is a nuclear membrane. When cell is dividing, this membrane disappears. So nucleus is not visible into the dividing phase. So it is only visible into the non-dividing stage. So it is visible in non-dividing stage, and it contains a chromatin network and the nucleoplasm. Actually, there is present DNA in the form of threads, and these threads are actually known as chromatin. When cell is in non-dividing phase, it will be called chromatin. When the cell will be in dividing phase, this chromatin structure will show this in the form of chromosomes. That is the distinction. So in the dividing cell, the nucleus disappears and the chromatin is replaced by the chromosome. When there is non-division, then chromatin will be visible, nucleus will be visible, but in case of dividing phase, nucleus will not be visible, the structure will be not chromatin, it will be chromosome. Chemical composition of nucleus. Nucleus is containing the hereditary material in the form of chromosome, and chromosome also it is consists of DNA, RNA, protein, and some enzymes including that. So nucleus contains the chromosome, DNA, RNA, proteins, and enzymes. Component of nucleus. Nucleus actually, for example, in this form, this is a nucleus. So the outer membrane is known as nuclear membrane, and there is present a mini nucleus inside the nucleus. This is known as nucleoli, and the region of the nucleus is nucleoplasm, and the structure present inside is chromatin or in the form of chromosome. So these are actually the four components. Nuclear membrane is outer, mini nucleus is nucleoli, region is cytoplasm, and the structure is chromosome. So introduction of the nuclear membrane. The nuclear membrane is surrounded by the nucleus is surrounded by the nuclear membrane, and this membrane is actually double membrane. So we called it also a nuclear envelope. So this is the membrane which covered the nucleus. So the nuclear membrane separates the nuclear material from the cytoplasm, as we know that. For example, this is a cell, and this is nuclear membrane. So this is the cytoplasm, and this is nucleus. So cytoplasm and nucleus, nucleus is separated from the cytoplasm by this nuclear membrane actually. The nuclear membrane separates the nuclear material from the cytoplasm. So the structure of nuclear membrane. The nuclear membrane is also known as nuclear envelope, as I said earlier. Envelope means that which contains two membranes, two layers, outer and inner. So it is composed of the two membranes, one is outer and one is inner. So these membranes are actually in this form. For example, this is inner membrane, this is outer membrane. This outer membrane is actually continuous with the endoplasmic reticulum, and particularly the rough endoplasmic reticulum present outside this nuclear membrane. For example, this is rough endoplasmic reticulum, and this is the outer membrane, and this is inner membrane. So outer membrane is continuous with the rough endoplasmic reticulum, RER, and the inner membrane is continuous with the nucleoplasm. So the outer membrane is continuous with the endoplasmic reticulum, and the inner membrane encloses the nuclear contents. That is the structure of nuclear membrane. Nucleopore. Nucleopore is actually, for example, this is the nuclear membrane. So nuclear membrane actually consists of the nucleopores and then so these nucleopores are actually responsible for the exchange of material inside or outside. So the outer and inner membrane are the continuous at the certain point forming the nuclear membrane. Actually, this membrane is continuously not attached. This is continuously present in the distance. These membranes are attached at a point where the nucleopore is present. So this is the point. Two membranes remain separate, but when there is a nucleopore, this membrane will join at that point. And the nuclear pore allow the exchange of material between the nucleus and the cytoplasm. Definitely, this is nucleus, outside is cytoplasm. So the exchange of material from nucleus to cytoplasm moves outside, or from cytoplasm to nucleus. It will be also facilitated by these nuclear pores. The number of nuclear pores varies depending on the cell type. The numbers are varies. Numbers are not fixed. According to the type of cells, these nuclear pores numbers are different. So the variability in the nuclear pore is depend on the type of cell. First is the undifferentiated cell. This human egg is undifferentiated. When the undifferentiated cell is consist of the 30,000 nuclear pores into its nuclear membrane. When we are discussing the differentiated cells, for example, the erythrocytes are actually the mature cell, or differentiated cell. Cytes mean differentiated cells. So these are also known as RBCs. See, the RBCs only contains a 3 to 4 number of nuclear pores in them. So this is the variability among the nucleopores. Function of nucleopore, each nucleopore has a definite structure. Definitely, they control the traffic of substance passing through them. As I have said earlier, this is the nuclear membrane, this is the nucleopore, this is nucleoplasm, this is cytoplasm. So the exchange of material moving inside out, it will be passed from the nucleopore, or the exchange of material moving from cytoplasm to the nucleoplasm, it will also cross the nucleopores. So the traffic substance passing through them is its function. Next is introduction of nucleus. Nucleus is darkly stained within the nucleus. For example, we stain the nucleus, and in nucleus we find this region lighter and we find this region darker. So this is the point. When we find the darker region, this darker region is known as nucleoli. It may be more than one into the nucleus. So nucleus is darkly stained within the nucleus, and it lacks any membranous bounded separating. So this is non-membranous actually. There is no membrane which separate it from the nucleus. It forms the rest of the nuclear material. The rest of the nuclear membrane material is not separating by any membrane. Numbers of nuclei. As I said earlier, there are many, maybe one or more nucleoli in a nucleus. So it's not specified that every nucleus will contain the one nucleoli. It may be more than one. So function of nucleolus, the ribosomes, ribosomal RNA, rRNA, is synthesized and stored into the nucleolus. Actually, as we say already, the endoplasmic reticulum contains the ribosome, and these ribosomes are the factory of protein synthesis. As we say, the ribosomes are the factory of protein synthesis. So the nucleoli is actually the factory of ribosome synthesis. So ribosome is made up of the ribosomal RNA. So these ribosomes are synthesized by this nucleoli. So structure of nucleolus composed of the two regions actually. There are the two regions. First is outer peripheral region, and second is central region. So the central region and peripheral region. This is central and this is peripheral. So difference is only the peripheral region is granular area. This is granular area composed of the precursors of ribosomal subunits. So as we know, there are the subunits which are the large subunit and small subunits. So these large subunit and small subunits are present into this peripheral region. It may be of 60s or 40s. So central is fibrillar area consisting of the large molecular weight with the RNA and rDNA. So it contains the RNA and rDNA. So this is the fibrillar region. Ribosome assembly, the nucleolus is the site where the ribosomes are assembled, and assembled ribosomes are exported to the cytoplasm via nucleopore. Look, we are describing this. For example, this is the nucleolus, and there is the formation of ribosome. After the formation of ribosome, this ribosome will travel through the nucleoplasm, and there is present the nucleopore. From this nucleopore, it will be moved out. For example, this is the rough endoplasmic reticulum. It will go and settle. So this is the assembly of ribosome and exported to the cytoplasm via the nucleopore. So these are the nucleopore. From the nucleopore, these ribosomes are exported out. So introduction of chromosome. Next we are discussing the chromosome. The chromosomes is the deeply stained basic dyes due to the chromatin material. When we stain the chromosome, its color is darker. So this is the deeply stained. During the cell division, the chromatin material convert into the darkly stained thread-like structures called the chromosome. As I have said earlier, if it is a chromosome, when there is non-dividing phase, this will be chromatin. This is non-dividing phase. When there is a dividing phase, there will be the condensation and the structure will appear in the form of these chromosomes, right? So this is a darkly stained thread-like structure called the chromosome. When we stained it, it shows the dark color like this. Chromosome structure we're discussing. For example, this is a chromosome and there is a central centromere. This is centromere and there is present the pair of arm. This is a pair of arms. So this is actually the structure. We also call it the chromatids, and we also call it the arms. So chromosome appear to be made of the arms and the centromere. Under the compound microscope, when we observe it into the compound microscope, it looks like containing the central centromere and the arms in the form of chromatids. Next is a centromere. Centromere is actually the place where the chromosome spindle fibers are attached during the cell division. This is a point actually. When cell is dividing, and there is centromere, and from the centriole there arises a spindle fiber, and spindle fibers attached to this centromere. So this is the point when there is a cell division. These spindle fibers are attached to a structure which is known as centromere at that point. Actually, it contains a kinetochore. So each chromosome consists of two identical chromatids at the beginning of the cell division held together at the centromere. So that is the structure. It contains two identical chromatids which are attached to this centromere. Composition of chromosome, actually the chromosome is composed of the two things actually. First one is the protein and DNA. Protein and DNA, actually we called it the histone protein. The protein present in the chromosome is known as histone protein, and actually these are the 8 molecules of proteins. 8 molecules of proteins. And also the DNA, this structure is actually in this form. For example, this is the 4 molecule of histone protein in the front. 4 will be present on its back side. Total will be the 8 histone protein, and there will be a DNA thread which will be attached to it. DNA is negatively charged. This protein is positively charged. That is why the opposite charges attract each other. So the composition is of the two things. DNA is actually present 40%, and this histone protein or the protein is present about 60%. Genes are located on chromosome and control the cell activities and transfer it from the generation to the next generation. Actually, we considered it, for example, this is a chromosome, and the DNA thread coiled it throughout its structure. When we open up it, there is present the nucleotides. These nucleotides are present throughout this DNA strand. If we say that from there to there is a sequence of nucleotide and we call it a gene. For example, the gene is A, then a protein will be formed. This protein will also be called A. So every gene contains the nucleotides, and nucleotides specify the sequence of amino acid into the further protein. And these genes are actually the inherited, which are transferred to the next generation. Next is the chromosome number in different species. So as every species has their specified number of chromosomes, so human contains the 46 chromosome. So it may be asked in the form of pairs, so pair will be the 23. Frog cell has a 23 chromosome with the 13 pairs, and the chimpanzee will have the 48 chromosome with the 24 pairs. As it is a fruit fly, Drosophila melanogaster, it contains the 8 chromosome in the form of 4 pairs, and the onion cells are the 16 chromosome with the 8 pairs. As it is the potato cells has the 48 chromosomes with the 24 pairs. As it is the garden pea, it contains the 14 chromosome with the 7 pairs. These values are very important for the MCQ's point of view. Actually, we are discussing the chromosome in humans. For example, this is the 46 chromosome with the 23 pairs. So there are the types of chromosomes. There are the two kinds of chromosomes. First is autosomal, and second is sex chromosome, or it is also known as allosome. Allosome. Actually, in humans, 44 chromosome or 22 pairs are of the autosomal chromosome, and the last one, 23rd pair, for example, XX in case of female and XY in case of male. This is the sex chromosome or the allosome, which determine the gender of next generation. According to the chromosomes, they are the individuals which are known as diploid and haploid. Actually, when the number of chromosomes are in the form of a pair, the individual is called a diploid. When there is a single chromosome, this is haploid. So the normal body of the cell have the diploid chromosome. Every body of the cell contains a diploid number of chromosomes. In case of human, every somatic cell will contain the 46 chromosome. When we discuss if there is a gamete, for example sperm or for example egg, so these are gametes. So gametes are the cells which contain only haploid number of chromosomes, 23, 23. So the normal body cell will have the diploid number of chromosomes, but the germ cells, for example, human sperm and egg, they will have the diploid n number of chromosome in number, which is half. And the human germ cells, for example, 23 chromosome, and Drosophila germ cells have the 4 chromosome. As it is, the Drosophila contains the 8 number of chromosomes, so the haploid number will be the 4. In case of humans, as the human contains the 46 chromosome, so there will be the 23 in the haploid numbers. So that was all about nucleus and its structure. Thank you. Allah Hafiz.